Good afternoon, everybody. I know I don't need a mic, but we're being taped for posterity. This is going to be the blockbuster of the spring break weekend. Will be the Ludcke chair talk. So thank you so much for coming and being like your students by sitting in the back and avoiding the front row. We are here to celebrate Dr. Markus Frederich, the Ludcke chair for this year. I sang you all his praises, sang his praises to you all back when the announcement came, and so I am just going to introduce him by saying that from quite literally the day that I met him, when I was a brand new dean, Markus was pretty much kind of like, nice to meet you, tell me what you're going to do about advancing research at UNE. And he's been in that mode happily and usually with a smile on his face ever since. It's such a delight for me as dean of the college and someone who's gotten to know Markus to see him receive the Ludcke chair. I know it's a very well-deserved honor, and these talks have been among the most interesting and fascinating and impressive that I've throughout all the things that we do here at UNE, and I know that Markus will not disappoint. Let me invite everybody to a very nice reception in the Bush Center immediately following the lecture, and with that I would like to introduce this year's Ludcke chair, Dr. Markus Frederich. Thank you.
Thanks a lot, Jeannie. Thanks for that kind introduction. It's truly an honor to be here, to be the one who was selected to give this presentation this year. And I want to start here with, now before I get started, so telling you a little bit of what's to come. I understand that this lecture is kind of, I'm expected to tell you a little bit about my past now. I got to the point that someone thought I was a worthy recipient of that award, and I'll certainly do that starting from the very beginning of time, and not completely here, somewhere there to where we are today. And I'll start here with that slide with the title, where the title is clearly stolen. No, I'm influenced by a nice book about crustaceans, which starts with walking sideways, and then based, of course, on the motion of those crabs. And then the book has a title of something like The Wonderful World of Crustaceans, but I thought my path through physiology is a nice one here. And all those images that are actually student works. I had a student who was pretty good in photography. She took that photo. Ilyana Fleffel actually drew that beautiful painting down here. Gwen made that nice dessert for a lobster bake, which we hosted for the lab, and the whole lab crew came up with the t-shirt logo right there in the middle. So I don't claim any having done any of that myself. Now, all those kind of award talks, they start with a quote from a famous book or a famous author to kind of frame the talk. And I want to do the same. And I'm starting with a quote which I actually have on the wall in my office, where I see it every single day. It comes from the famous book of Kevin and Hobbes. I asked myself, what on earth am I doing here on this beautiful day? And this is the only life I got. And when I look at that, it reminds me to think once in a while, is what I'm doing really what I want to do with my life? And I'll pick that theme up throughout the talk again and again, so I don't lose track of that. But for showing you that I don't only read comics, I want to use another quote from a famous physiologist, Knut Schmidt-Nielsen, who starts his autobiography by saying, "It has been said that the primary function of schools is to impart enough facts to make children stop asking questions. Some with whom the schools do not succeed become scientists." And obviously I was a school failure here. But this kind of asking questions is really something which goes straight through my career as a scientist and also as a teacher. And I'll pick up that theme throughout the talk again and again. So, the whole story started a while ago when little Markus grew up in a little village called Großumstadt, somewhere in the middle of Germany, this quaint little village with this nice and old historic town center. And old here is meant in a European way, not the American way. That town hall is built in the 1590s. And there are about 10,000 people living around, so kind of uneventful place on the planet, except once per year there's the annual wine festival, which combined with a not enforced legal drinking age of 16 led to certain distractions. But despite those distractions, yes, I managed to graduate from that kind of scary looking high school and was then faced with the even scarier question, what should I do with my life? The simple answer was, I had no idea. So I took my backpack and traveled by bus and train through Europe down to Turkey and then by boat over to Israel, where I worked in the kibbutz for a while, and that by accident coincided with the beginning of the first Intifada. And then traveled by bus. I wasn't involved. And then traveled by bus and train up and down the Nile River in Egypt. And when I came home after a few months, I still had no idea. So I started working as a worker at Merck, this pharmaceutical company. I had this brief idea of becoming a journalist. So I started writing for a local newspaper. That didn't last. No, yes, after high school, yes. And then became a truck driver for a while, and then started working as an ICU nurse. And that kept me busy and excited for a while. And learning about all those diseases and mechanisms and organs and failures of them. And then also all those fancy machines, that was really exciting. But I also learned over the years working as a nurse that I don't really care that much about those patients. But then medicine without the patients is biology, right? I found my true calling. So I went to the Technical University in Darmstadt for studying biology, and just because I was involved in quite some curriculum development and core curriculum discussions here, I want to briefly tell you about the curriculum at that school, where I didn't even list all the cognate courses here with chemistry, physical chemistry, OChem, physics, all that kind of stuff. We basically had a general ed requirement of what we would call here major plus. I needed a major, which was biology, and I needed a minor, as the plus, and I took geology. Therefore, 100% of all the courses were in the sciences, which allowed me here to do my explorations of biology, botany, geology, zoology, taking advanced courses in pharmacology and so on, and learning all those critical thinking, problem solving and so on skills, taking courses that I really cared about. And I had a blast. I'm not telling you that we should adopt this as a core, but only for saying, even this one works. What I didn't do there is undergrad research, because undergrad research didn't exist. Actually, at that school, kind of as undergrads, we were basically non-existent. They didn't care much about us until we became then grad students. And so I did a master's thesis on some ants, which got kind of damaged most likely by the Chernobyl disaster, and taking up some of that radiation made not only typical males and females, but also those kind of sexual interforms, where some of them had testes and ovaries at the same time. Some of them were lacking some eyes here, there's one missing up there, or they had those strangely formed antennae. It was my first real exposure to research, and realized, yes, this is great, I want to do more. But this was not really asking questions, this was mainly descriptive. It was describing what was there, and that was it. Everybody was happy with that. But there was no diving into the details. Why did that happen? What caused that? I realized, okay, so I got the straight zoology, that's not the right thing. Physiology, that's where we asked those kind of why questions and mechanisms. So I got into a PhD program at the Alfred Wegener Institute in Bremerhaven, which is up here at the North Sea coast, where I did my PhD work, and then also spent, as part of that work, a few weeks at the Station Biologique in Roscoff, here at the northwestern tip of France, and spent a few months down here at the Universidad de Magallanes in Punta Arenas, the southern tip of South America. That was kind of the study abroad component. And here, the whole project was really framed around a cool question, where for decades, researchers who went to the Antarctic figured out there are all kinds of critters down there, including crustaceans, except all the crustaceans which you find around the Antarctic here belong to the group of the shrimps, those natansias, they were called in the older systematics. And that otherwise worldwide successful group of crab-like crustaceans, reptansia, is completely absent down there in the Southern Ocean. And no one had any idea why that is, and they tasked me to get another idea. So in that context, I looked at various different critters, and for example looked at general temperature stress physiology, and remembered my days as an ICU nurse where all the patients were wired up to different beeping machines. I thought I can do that with some crabs. And I attached a heart rate sensor to those guys and drilled some holes in there and stuck, like here, an oxygen probe right into the heart and some flow meters to the different blood vessels. And we could measure ventilation, all feed that into a computer and record that online while we're heating up or cooling down the temperature. That led to data like this. We have temperature from 0 up to 40 degrees, for example, heart rate, which is low at low temperatures. It goes up, levels off, and crashes when it gets too warm. Ventilation, low at low temperature, it warms up, gets higher, levels off, and they crash again. And then the oxygen in the blood, which kind of is important for making all the energy for being alive, is low at low temperatures, goes up, plateaus, kind of nice curve shape, goes down, and gets close to 0 at the high temperatures. At that time, the kind of big deal in physiology was identifying what was called critical temperatures. Those critical temperatures were temperatures at which animals switch to anaerobiosis. They start making energy without oxygen. Like when you're running too fast and next morning your legs feel tired because they accumulated too much lactic acid. And those critters do the same thing, accumulating lactic acid even without running, just by being exposed to very low and very high temperatures. And that was nice. It wasn't shown for crabs before, but it was kind of at the extreme ends. At those time points we ground them up and measured lactate and went up. But what we also saw is that way before the critters reach those critical temperatures, there's more happening, like right here, where we hit the maximum heart rate, maximum ventilation rate, and at that time, oxygen goes down. Down here, more hidden in the data, easier to see up there, where kind of the oxygen drops off, there's another kind of break point. And physiologists love breakpoints. If something is happening, then we can come up with whatever kind of ideas of what that might be. So okay, we need a name for those new breakpoints. Now remembered my ecology lecture where I learned about the law of tolerance from Shelford in the 1930s. Where he said for any kind of critter, can be a human, can be a crab, can be a bacterium, pick your favorite one, there is for any kind of abiotic factor, temperature, pH, salinity, whatever. There's a certain range where this abiotic factor leads to an optimum performance of that respective critter, and in the trends where they don't do so well. And then he called that optimum and pessimum. We said, okay, we can use that. We can say those critical temperatures where the critters switch to anaerobiosis. That's the transition to the pessimum range. Because if they're exposed for longer periods of time to those high or those low temperatures, they die of lack of oxygen. And that kind of defines the physiological tolerance range. And then we said, but before they reach that, so if we go back here, it kind of gets worse right there. And getting worse in Latin means paius. So we inserted that new paius range in a model and termed those new thresholds here paius temperatures. Those are temperatures where suddenly they leave this optimum range and life gets worse. And it turns out for all the crustaceans which we looked at, those paius temperatures nicely described kind of the mean minimum and mean maximum temperature what the animals see in the environment. And that kind of is then the range of maximum performance. Then we put that in a publication and moved on to do other stuff. And it turns out actually that idea really caught some traction, and by now it shows up here in the textbook which I use for teaching my animal physics course. We have here critical temperature, right there is the paius temperature. And tell the students that if they are frustrated they have to learn another term, they have to blame me. So I invented that. And they're all in awe. And that, from that moment on, I can tell them whatever I want, they believe everything, which makes teaching so much easier. I will not comment on that. And then this concept is by now also included in the most recent IPCC report on climate change here, where in the chapter on the ocean, the cryosphere, the authors actually describe this modeling optimum payos critical threshold with some modifications in terms of how it changes in the life cycle of animals, for then actually describing how animals will be affected by climate change. So that was a fun story, which I'm going into that detail here because I'm still working on some of those aspects today, so we'll get back to that. For another part for that story, I spent some time in Chile. That's our drive here to one of the field sites. That's the Marine Science Center down in Punta Arenas. Yes, it's cold. There's some ice, which is kind of nice. If I wouldn't have fallen in love with crustaceans, penguins would have been the next thing. They were just hopping around everywhere, and they're adorable. Those are those Magellan penguins. And then hiking in Torstolpine National Park is just spectacular. That was a neat time. Good. Back to some data. What I did down there is kind of drilling deeper into what is different between those shrimps and those crabs in terms of getting to the Antarctic and not getting into the Antarctic. And instead of showing you all the different detailed data, just show you what we found. The basic conclusion of all the work was it comes down to magnesium. So it turns out magnesium in the bloodstream of invertebrates leads to anesthetizing them. And that anesthetizing by magnesium is more potent at low temperatures. So if you combine high magnesium and low temperatures, the animal falls asleep and dies. And for some other reasons, it turns out those shrimps have low magnesium levels. They can stay active at cold temperatures. Those crabs use a different mechanism for regulating magnesium. They have high magnesium levels, and they just fall asleep when it gets too cold. They can't make it. Now, in that context, while I was working down there in Punta Arenas, I stumbled over those cute critters here, Helicosinus planetus, small crabs with the size of a dime. They're crawling around there, and they live all around the Antarctic. They're well known, just I didn't know them at that time. But I was kind of poking every single crab to get blood which I could get my hands on to measure magnesium levels for kind of supporting this whole magnesium hypothesis. When I measured magnesium in those critters, I found those are the crabs with the lowest magnesium concentration of any kind of crab. Pretty close to the magnesium level of those shrimps in the Antarctic. And I did some other experiments to test for temperature tolerance, and found they're amazingly cold tolerant. And I thought, if low magnesium allows those critters to live in the Antarctic, or higher magnesium excludes them, then this one, which has the lowest magnesium concentration of all those crabs, if there ever is a crab to make it into the Antarctic, this will be the one. We snuck that bold prediction into a paper, and then I forgot about that and went on to do other stuff. And then, I think 3 years ago, I was sitting in my office, my desk with telephone rings, and a buddy of mine calls, a former fellow grad student who is now a professor in Great Britain, and he says, "Remember how you predicted those critters in the Antarctic? Guess what? We found them." Now, it turns out a friend of my friend was also a marine scientist who got sick and tired of science and became a naturalist on those cruise ships which go to the Antarctic. He was down with tourists in Deception Island, right here, the small crater island, which is heavily frequented by those cruise ships because it's nice sheltered caldera and warm water for the tourists to go swimming. And then that guy, while the tourists were taking the photos of the penguins, was kicking up some rocks on the beach and found a crab. Actually, this one, that's the one. And he remembered from his marine science days, there was something funky about crabs in the Antarctic. So he picked it up, dropped it in a jar of vodka, and took it home with him and sent it to my buddy in Great Britain, who, when he opened the shipment, nearly fell off his chair. Because that is the very first reported crab in the Antarctic. And then, even more important for me, my buddy remembered that I actually predicted that this would happen many years ago. So we wrote up a neat little paper on how those ones make it into the Antarctic. Here we go, where I put together this figure which shows magnesium concentration of a whole range of crustacean species. Well, we have up here all those crabs which live all over the world, warm and cold temperatures. Down there are the Antarctic shrimps, low magnesium, lower than 12 millimoles. And here is that Carcinus, the one which they found down in the Antarctic. And it fits right in there. And while I was putting that figure together, I realized, wait a second, the next crab in line in terms of being down there is the green crab, Carcinus maenas. The green crab lives out there in the river and everywhere, worldwide. So we actually, here we go, put that figure in that paper where we show in blue here the distribution of that Carcinus, which now made it here into the Antarctic. And in the same place, we also find green crabs. So we snuck in that paper the prediction, the next one will be the green crab. Now every day I'm sitting in my office, in my desk, and wait for the telephone to ring, for someone to call me and say, we found it. Didn't happen yet, but probably takes another degree or two of warming, and then we'll get there. The reason actually why that is a big deal is, it's not that, okay, now they're there. The Antarctic is a very special ecosystem in terms of it evolved over millions of years the complete absence of any shell-crushing critters. Therefore, all the bivalves and other shell-bearing animals down there got over the millions of years thinner and thinner shells. If you now have those critters with claws in there, that's like letting the fox into the henhouse. It will be a bloodbath. And it's not a question of if that happens, it's just a question when that happens. It's just another degree or two and they will be there, which will change the ecosystem completely. So I actually use this story in my teaching when I tell students about how the scientific method works. And this is the typical textbook version. Yes, we ask a question, we do some background research, then we come up with a hypothesis, we test it with experiments, we analyze our data, and we publish that somewhere. In this case, yes, I was following this magnesium question, a magnesium hypothesis, and along the way made some other random observations. I added some experiments just to frame those observations for being able to sneak it into a paper. Then snuck it in there with a bold statement. Then I waited 14 years. And then I used the word serendipity. I like that word. More honest would be pure dumb luck. Which then leads to, okay, we report the results again and remind people that I predicted that long ago. Here we go, that's how it goes. So that is for the students then to see, okay, that is not really how science works. There is way more involved, which that one plays a big role in there. Okay, so that's all kind of neat branching off of that original PhD work, and if we go back to that thing, yes, I finished that and they awarded me a degree, and then I was faced with the question, and now? And my advisor at that time offered me a great postdoc position. So I could do a project on natural products in the Antarctic, which would have included several trips with the RV Polarstern to the Antarctic, and then working with the pharmaceutical company BASF on identifying some of those biologically active or medically important compounds. And then somehow I didn't listen to Calvin Hobbes with the fun part. I really thought, I read the contract actually in detail, because that being one of those industry positions, they had a long contract with lots of details which were very scary. And I also thought, if I do this, after 2 or 3 years, I'm still a marine biologist. And there aren't any jobs for marine biologists, at least not in Germany. And I knew, okay, then I have 2 or 3 years of fun and then I'm doomed. So instead I said, okay, let's follow the money, funding. And funding is there for medical research. So I thought, okay, I can do that. And then stumbled over this little job ad here for a position in cardiac metabolism work at Harvard. And I did hear about that little school before. But for everybody who didn't do that yet, it is a very interesting feeling to hop on a plane with a one-way ticket to a country where you don't know much about, to a city where you've never been, to work at a school where the name alone makes you feel very inferior. Oh well, they let me in. And I did some work on rat and mouse hearts. So the basic idea was we took those rat and mouse hearts out of the animal and put them up on one of those perfusion apparatuses, and they start beating for the next 5, 6, 7 hours. And then we can stick that whole setup into an NMR spectrometer, and then we can measure the energetics to see how much energy they have if we do different things to it. That is important for various different disease models, and was a fantastic time doing that. But I also learned I don't want to do that for the rest of my life. I really want to get back to marine science, where really my passion was. So I applied to schools all the way up and down the East Coast and West Coast, and found this little school in the woods which has this pretentious name, University of New England. I thought, okay, I can go there for a year or two and see where that works out. And obviously it did. Well, then I was kind of faced with the problem or a question of how do we set up a research program here. That was at a time where research wasn't that seen as that important. I remember well one member of the search committee, I will not reveal who it was, told me, you will never be able to set up any research here. That's not what we are about. The other member of the search committee said, he was a little bit more positive. He said, if you ever want to fulfill those research requirements to get tenure, you need to do teaching research, because you'll teach all day long, and if you can combine that with your research, that's your only chance of making it. I said, thanks, great advice, and I ignored it. And then remembered kind of what I learned earlier. And during my PhD work, I learned this whole model here, which comes down to, at the end, thermal tolerance is limited by energy, ATP. Critical temperatures, Peyer's temperatures, and so on. And I also learned during my postdoc time about an enzyme called AMPK, and without going into all the details, the basic role of that enzyme is to regulate ATP. And at that time, what we knew about AMPK is it exists in humans, in rats and mice, in the fruit fly, and in yeast. Because that was what people cared about. Nothing in there, but we knew it's highly conserved during evolution and regulates the ATP. So I thought we could combine those two things and say, is AMPK actually involved in temperature tolerance? Because at that time, all the invertebrate people never heard about that before, because all the medical field published in that. There was a wide open field where I could slowly work my way towards that without being scooped by someone else. So I got some crabs from the lobster guy across the river, extracted some tissue, got the DNA out of it, ran that through a fancy sequencer, got data like this, ran those data through a database, and yes, there it is. Steve is laughing. This slide summarizes my first 2 years at UNE. So now we knew, okay, yep, they have that. We can see, is it activated by temperature? So we can incubate those crabs at different temperatures, then, yes, find those different thresholds and see what is AMPK doing here. There are some of those data where you see the rock crab and heart rate, yes, increases with temperature and then it crashes. We've seen that before. Here's lactate, indicating whether they work on aerobic or anaerobic metabolism. Here they switch to anaerobiosis at a certain temperature. Yeah, we looked at those heat shock proteins, which, like the name really tells you, they're involved in heat shock. And AMPK here gets activated by heat stress. That was great. So we can put that into one of those fancy models here where we have, over the years, the water temperature in the Gulf of Maine. And we can put at those different temperatures here the respective parameters which we measured, like AMPK goes up right here at a time where those critters get slower, or Peyer's temperature. Up here is where the traditional heat shock response kicks in. And until then, everybody looked at those kind of parameters, heat shock protein, lactate, and so on. And we made the case that the Peyer's temperature is more important, and AMPK is a great indicator for that threshold. And here's Michaela, who's presenting that work at the EB meeting in Washington a few years back. She also became a co-author on that paper. Michaela then went to the dark side and studied medicine here at COM and is now a physician here in Saco. I don't hold that against her. Then we moved on and said, okay, is that true in different crustaceans as well? Or is that kind of a fluke that only happens in those rock crabs? So we used then not only the rock crabs, but green crabs and lobsters, and did similar experiments where with here AMPK goes up in rock crabs and lobsters, lactate goes up in some of them, heat shock response in some of them, yes. We also measured activity, and without going into great detail every single data point, the green crab, we couldn't pick up any AMPK activity. We couldn't figure out that pH temperature. So we again came up with a fancy model to describe that. So okay, for the rock crab and the lobster, everything what everybody knew until then applies. There's an optimal range of the temperature, then we can define a pH threshold. There's a nice pH range where it gets worse, critical temperature, and they die. The green crabs, they go fast until they drop dead. There's kind of no transition from perfect to dead. So I said, okay, AMPK seems to be a great indicator for temperature stress, but it doesn't apply in green crabs. We put that in a publication where Stephanie is a co-author, and there she was presenting at the EB meeting in Anaheim, California, and got an award for that. And she's now doing also some medical stuff, and I also don't hold that against her. So all this work and some others then led to a review paper, which I wrote together with my good friend Ina Sokolova from the University in North Carolina in Charlotte, Rita Bagwe at the Alfred Wegener Institute, and Alex Sakotin from the Zoological Institute in St. Petersburg in Russia. We kind of combined this, this whole pH critical temperature story, now got this fancy name where someone else came up with the oxygen and capacity limited thermal tolerance hypothesis. And someone else came up with the idea of a dynamic energy budget. We put that together. We say, under normal conditions, an animal, normal meaning everything's fine, needs ATP, energy, for some basic processes, storage, reproduction, growth, activity, maintenance. And you can fit your whole biology to those basic parameters. And all this covered by aerobic metabolism, lots of activity, then the optimum range. If we now stress them, then maintenance cost goes up. They don't store anything anymore, and there's less energy for growth, sex, or anything else. And that's still covered with aerobic metabolism. Now in the pH range, they're struggling. If we heat them up even more, we'll ignore this for now, then everything the metabolism is doing is just trying to maintain a stay alive, and they switch to anaerobiosis, they're in the pessimal range, and they're on the way to being dead meat. And this is kind of a beautiful general roadmap for the next 20, 30, I don't know how many years of science in my lab. Not that long? We'll figure that out. Because now we can ask all those questions. What is happening from here to there? Or what regulates that? And so on. And it seems other people were excited about that too. It actually took only very few years until that figure got picked up by George Somero in his most recent edition of the biochemical adaptation textbook. So now I can even use that on my students and they can't complain. Good. So this is, to this day, kind of a roadmap of how I see the world. Through some serendipity, other coincidences, then I got more interested in green crabs. And they are the scary invasive species. They come here from Europe, and they made it all the way to all different continents except Antarctica. They moved into Massachusetts exactly 200 years ago. It was a big birthday this year. Australia, South Africa, everywhere. And they become this amazing pest by coming in huge numbers, like here shown. The clam digger took this video where they pull up some of that mud, and underneath there are hundreds of those critters destroying the ecosystem. They're eating the clams, they're destroying the whole clam harvest. They're a real pest. They're that important that even Governor LePage started a working group on trying to figure out what could be done about those green crabs. So they're out there. And so we've follow the newspapers, there's again and again articles like this where different towns come up with different strategies of fighting that invasion. At this point, nothing works. Now, what we got interested in is those crabs change color in their life cycle. And they can be bright green, they can be dark red, and everything in between. And the basic story is that those crabs start out as little larvae, and then they molt, and then they're green, and they molt again, and they stay green, they molt again. So they have this mode where they're really focused on growing quickly, and they just stay green. At a certain age or size, they can also just delay the next molt, and then move towards becoming reproductively active. And then when they delay the next molt, they actually get redder and redder and redder. And what was known by lots of papers in literature here over the years, and I overlooked that work for years until someone reminded me, that those red morphs, through all those different parameters, hypoxia tolerance, salinity tolerance, and so on, are less stress tolerant. The green ones are way more stress tolerant. I thought, okay, that includes a ton of questions. So, for example, put them on the treadmill where we can measure how well they can actually run. This is going to be the walking sideways part. We can change temperature, salinity, speed, oxygen availability, and so on, and see what they do. In this case here, we exposed them for 24 hours to hypoxia, low oxygen, nearly no oxygen. And the green ones do after 24 hours without oxygen just as good as in the beginning. That's like you holding your breath for 24 hours and hopping in the treadmill. They do better. The red ones don't do so well, but still can run. We can also measure lactates, some other parameters, and find there are clear differences between those red and green morphs of those crabs. We can do salinity stress. In this case, Anthony built this contraption by having some automated valves with timers and everything. We can measure, we can change in the incubation tank salinity from full strength seawater to brackish and back and forth and back and forth. And can have the crabs in there, and then we poke them every time when the conditions change. And we can draw blood samples. And we find, yes, the iron concentration in the blood also oscillates with the salinity here, but it oscillates more in the red ones, meaning the green ones are better iron regulators. We can have them run on the treadmill. After all that stress, the green ones run way better than the red ones. Then we can get to the mechanism. How does it work? And this is a simplified model of the iron regulation mechanism here in the gills. Those crabs actually use their gills for regulating ions. And I don't go into all the details, I do that in my physiology class. The point here is there are lots of individual transporters which move ions in and out, and we can measure basically every single one of them and see what happens when we do bad things to that crab. And then we can, for example, after exposing them to different salinity conditions, measure those different transporters in there, the proteins, and can quantify which one gets upregulated, which one gets downregulated. We can also quantify the gene expression. Before you get a protein, you need a gene which is transcribed. And then we can find which of those genes is up or down regulated in those different conditions. My favorite enzyme, AMPK, doesn't play any role. That made me sad, but I got over it. And we found that some other of those targets here are clearly responding to that stress differently in red or green ones. And then again, we can put that into a broader model for understanding what we see here, where we have, while the animals are growing, they are not reproductively active. They're green. They have a lot of energy which can be invested into iron regulation. They have all those mechanisms by specific parameters to deal with low oxygen stress, and overall are more stress tolerant. Versus when they move into the reproduction mode, less energy goes into iron regulation, less energy goes into the anoxia response, and they're overall less stress tolerant. Or we can put it into this broader dynamic energy budget. We can say an average crab under normal non-stressed conditions uses ATP in certain amounts for those 5 categories. And if they turn red, more and more energy goes into reproduction. They don't grow, and less goes actually in maintenance, which leads to reduced stress tolerance, which now again leads to dozens of questions. Why this, why not that, and how is that regulated, and so on. So that can keep us busy for a while. And after more than a year of battling with the editor over the shades of red and green in the figures, we finally got this published this year, where well-deserving co-authors, here's Wing Q, who's actually the son of my postdoc advisor, and then Gwen and Anthony, who are showing off their work here at different conferences, and they're also moving on for doing cool stuff. Now, through working on those crustaceans, I stumbled over the fact that there seems to be a difference between where those critters live. And so like I said before, in the early 1800s, green crabs came over here to the US. There was actually a second invasion from Northern Europe into Canada, and then they moved on from there. But that led to genetically distinct populations of green crabs. They're different. And now we can ask questions. Are they different in terms of how they behave or what they do or they not do? And then here, for example, is Lauren, who's measuring the three-dimensional structure or size of those crabs. Lauren graduated last year. She's now a fisheries observer, one of those fishing vessels in the Bering Sea. But she spent hours on kind of digitizing specific landmarks on that crab and get kind of the XYZ coordinates to then assemble those into those wireframe models we have here, the three-dimensional structure of the crab back front part here. And then we can do some fancy statistics and try to figure out, are they actually different in their shape? Because they looked different. And from some first analyses of the data, it seems that the crabs from Iceland are actually a bit narrower and longer, and the ones from Canada are wider, and so they are clearly different. It's kind of neat, and we're still working on the analysis. When I say we, I'm lying, because I know nothing about how that actually works. Joe is the expert on that one, and he'll help us figuring that out. We can then expose those crabs from different places where my grad students drove up to Nova Scotia, Newfoundland to collect some critters there. I brought some crabs home from Iceland. The maenas were easy to get. And then we can expose them to different temperatures and different salinities, and then, for example, run them in the treadmill. And without, again, getting lost into every single data point, for example, one of the things which sticks out is the Icelanders do very different from all the others. Now, those Icelandic crabs, they are kind of the native population. The main Nova Scotia, Newfoundland ones, they are the invasive ones with a different genetic haplotype. Now we need to sort out what that actually means and how we can explain that in more detail. Nice thing about the Icelandic ones is the Icelanders seem to be so proud of their green crabs. They're the only ones which I'm aware of who put a green crab on their currency. That's on the 50 krona coin. I brought a whole bunch of them home with me. What we also learned while working with them is that there's a very different aggression level in those crabs. And here's a typical low aggression green crab. When you poke them in the water, they kind of tug in their claws, or if you get too close, they kind of try to push you away. But that's all what they're doing. Oh, here's the chicken. Look at that guy. He just runs away. Okay. Here we've got a medium aggression green crab. They're kind of spreading their wings, claws, and going into this aggression posture. They try to stand their ground, but that's about as far as it gets. You can touch them and annoy them, and they threaten, but that's all you get out of them. Here we go. There again, nice aggression posture, good. And then there are a few where we lost our finger before. You stick the camera in the water and here they come. Here again. Yes, here come the claws and get it. Here we go. Doom. Yes. So they're clearly different in their level of aggression. Then we can test dozens of those crabs, got to score them, how many from the different population are how aggressive, and we find here the ones from Nova Scotia. Very few have this shy stage 1 aggression. Most of them were just going at us. The maenas, they're all laid back and they're running. Yes. The maenas, they don't do much. Not a single one attacked us. And the ones from Newfoundland and Iceland are somewhere in between. Now, of course, question is, why do we care? Those green crabs, for example, destroy eelgrass beds. Those eelgrass beds here, they're everywhere. This map which shows in Casco Bay, all in black, that's all eelgrass beds everywhere. Those beautiful underwater meadows here, which are important nursery grounds for fish and other species. They're an important carbon sink, also important context of climate change. They protect the coast against erosion and everything. And once green crabs move in large numbers, they wipe those eelgrass beds out. Now I thought, okay, we have here crabs with different levels of aggression. Let's see what happens if we expose them to eelgrass. So we built a little eelgrass bed, right there it is, big pool, that's inside the eelgrass bed. So we planted all that, and my grad student for setting it up was busting his, not his butt, he was actually literally busting his knee while carrying all that stuff, but he got over it. And then we let crabs from those different populations into that artificial eelgrass bed and counted how much destruction do they actually cause. In this case here, those freight blades, if we compare here, maenas, they rip out a few. The ones from Nova Scotia, they rip out way more. And here comes the bad news. The ones from Nova Scotia are coming to us. Right now here, all those maenas are living down there, up there, the ones from Nova Scotia, and driven by the ocean currents, those haplotypes, kind of those genetic kind of crabs from the area in Nova Scotia, they're moving south, and they come here more and more and more. In a few years, we'll have mainly either these ones or hybrids between those ones. So most likely, way more aggressive crabs than we have right now. And green crabs are already now a big problem, so in the future they might be way more problematic. We'll find out. I'll be here to watch that. Yes, talking about watching that. We're also running for many years now kind of a survey for 2 invasive crabs. Here's the green crab, and we're going out the Biddeford Pool every month throughout the year for 6 years now. And then we count every single green crab and also every single Asian shore crab which we find there. So it turns out green crabs are here for 200 years. Asian shore crab came into Maine about 15 or 16 years ago. And when I started this monitoring project, my thought was, great, the literature says once the Asian shore crab moves in, it displaces the green crabs. So I was planning on kind of documenting how these guys take over and displace those guys. Turns out they didn't read the right literature. They didn't displace the green crabs. They live happily side by side, which shown down here, all the same. That we can also measure when are they gravids, when do they carry eggs, when do they reproduce, when are they how large or small, and can put together a whole population analysis, which we're actually working on publishing right now. Now, more recently, I thought, okay, now we have all this multi-year data of those critters, what they do in their transect, and we know when they're gravid, when they carry the eggs, and then they release those eggs and they form larvae, but we don't know anything about the larvae. So I bought a fancy instrument, this flow cam here, which costs more than my annual salary. NSF was kind in funding that, which is called an automated microscope, which, yes, came out of the Bigelow Labs. They now have their company in Scarborough. So you feed in your sample up there, down here is the microscope, and then a pump which sucks the sample through, and in there is a camera which then here takes photos of every single particle which runs through, and within a minute or two you get hundreds of different critters showing up there, and then you can tell the computer to sort all those into shape and size and color and taste, no, not taste, all those other different parameters, and then within a few minutes you have a whole analysis of the plankton population, which that's the kind of science I like to do. So otherwise analyzing that by hand would take a week or so. So now we'll extend that to figuring out what's out there, and so far we got some beautiful shots of, those are some barnacle larvae. Here we have some jellyfish larvae. Those are actually some eggs from some snails, some copepods. Here are the crab larvae, and over time we'll hopefully build that great long-term data set of what's out there. While working on that, we realized being reliant on a boat every single time for getting that plankton tow is pretty expensive, and doing plankton tows near shore is kind of a problem with a big boat. So we actually built a small remote-controlled plankton sucker. That is a boogie board here, which has two thrusters underneath, a box with the electronics and the battery on top. And then we can, with a remote control, just drive that out there. There's the control. And then we can turn on here this suction device, which is another propeller in the back, which sucks water straight through. We can measure how much with the flow meter and catch all the plankton in that net. And up there is Ariella, who built that thing. She's sitting back there. She's being filmed right now here by Sean, and actually 2 or 3 seconds of that footage made it into the most recent UNE commercial. So that's Ariella's 2 or 3 seconds of fame. And then we can actually go out there. In this case, it was our first test run of the boat right in front of Ram Island. We can drive it through the water, turn on that suction device. Here we go. It sucks in all the plankton. Then we can pick it up again and run that plankton sample through the flow cam, and it'll tell us immediately about those hundreds of cute little critters which we just removed from the ocean. So all those stories kind of fit into a few of those general questions. How do marine invertebrates allocate energy during stress? And how does it differ between species and populations? And how does it affect the tolerance in various species? We can look then at invasive species just because they make a good story, and we can always go back to kind of this theoretical framework for fitting our broader descriptive data into a mechanistic understanding, which hopefully will bring us forward step by step. Now at the very end, I want to talk about the other part of my life, what I'm doing here at UNE, and that certainly is teaching. And I think all of us are well aware of many of those students who are sitting there and wonder after a few hours whether the teacher said anything important. Over the years, while teaching more and more, I started asking myself more and more whether what I'm saying is actually anything important, in terms of whether my teaching really leads to their learning. And more I really thought back about that quote which I used earlier from Knut Schmidt-Nielsen here, this dumping data or facts on students, and especially in the sciences, we're heavily guilty on that. We expect them to memorize so many random facts, it's kind of scary. And more when I design courses or think about my lab activities, I wonder, are those questions where we know the answers? Do we need to ask them? As in most of those lab activities, yes, we tell the students a great story, what they will be investigating. We know exactly the answer. If we don't get that answer, the students screwed up. And over the years, I thought that's kind of boring. And I more and more start thinking about, okay, what can we do for having the students answer questions where I don't know the answer? Which means we build real research into those courses. And that's kind of scary as a teacher sometimes, but it's also fun. And that kind of thinking was influenced and is still influencing 2 large NSF grants, which I got for some teaching work, where a few years ago with Larry Fritz, we got the Synergy grant, where we actually provided scholarships for research active students. We recruited them, supported them, they got active in some cool research, and that was kind of built on the honors program, which at that time was kind of implemented and pioneered by Greg Zog. And building more and more of that research into their curriculum kind of then started also increasing the undergrad research endeavor in the college overall. And it led to creating an undergrad research conference, the NERDS conference, which over those 6 years we had actually ran, brought overall more than 1,200 students from 40 different colleges to UNE. They were all showing off cool research. And our uni students were really shining in comparison to many of the others. And then I'm currently working on that turbo grant, which I got together with Steve Seaman, Ursula Rosa, and Leah Beimers, where we use kind of the Saku River as our biodiversity observatory. And in this project Turbo, we kind of implement in different classes or fund already existing activities which are real research experiences. We do kind of place-based education. We use the Saku River, and we define the Saku River as broad as we need to include as many people as possible. And by now there are hundreds of students in many different departments who are doing those kind of turbo modules. Actually, just this week, more than 300 bio students complete a turbo module on DNA barcoding. In the marine science, they're in various different classes. ENV has several of those. Chemistry did a turbo activity on water chemistry. James Quinlan's database development class is now working on actually developing a database for those data. And so by now, many hundreds of students actually did real research projects in their classes where the instructor didn't know the answer in the beginning. So there was good questions on DNA barcoding, intertidal communities, wetland flora, genetics of invasive grasses, plankton surveys, and more. So when I look at that little piece of paper on my wall and ask myself, is that the right thing I'm doing here? I think yes, whenever I'm able to combine this kind of excitement for research and really infusing that directly into the teaching, and that's really time worth spent. I'll end right here, acknowledging a few people who were important along the way in shaping what I'm doing and how I think about the world. And of course, those kind of acknowledgement slides at the end, they're only there for showing the audience that the presenter knows a few important people. Way more important is actually this whole army of undergrads, mainly in this slide, there were 2 or 3 grad students in there, who were involved in all those projects and who did amazing work and who learned a ton about crabs. Yes, most of them don't care about crabs anymore, and they shouldn't, because they moved on to become physicians, government worker. One even became a librarian, and so on. But they all were able to apply what they learned in different courses. They all learned problem solving, critical thinking. They learned writing by writing proposals for summer projects. They learned presenting that work to other scientists. And I think they got a lot out of it. All that kind of work is insanely expensive, and I was fortunate to get some money from various different organizations. NSF, through their iOS program, paid a large research grant, which allowed me to actually get started. The education section of NSF funded 2 large grants. The major research instrumentation group, they funded the FlowCam. Barry's EPSCoR grant helps me once in a while with money. I had several students who were paid for their summer through the American Physiological Society. I spent 2 summers up at Mount Island Biolabs in Bar Harbor, and they paid me for being there. Main Sea Grant paid for a project on clams, which I didn't mention. And then various different offices at UNE, like the Dean's office, where we still had those Dean's mini grants, or now through travel money, or the VPR grants, or the Marine Science Center. They're amazingly helpful in keeping me busy and keeping the students busy. I'll end right there and open that up for questions. Yes.
I sort of agree with you that student research is really a place where they learn so much. But can we turn curriculum into, say, project-based learning? I mean, so chemistry probably might want to turn quick.
Right.
But there have to be some original courses.
Yes, that is a great question, and I think that is one of those big questions throughout the pedagogy literature. How much do students actually need to know? And I grew up taking lectures which was just dumping facts at me. And it kind of seemed to have worked. And when I design my courses, I'm always caught in that big question, how much of that content, which I think, yes, they should know, can I cut out for creating enough time for doing those kind of project-based approaches? I don't think there's a perfect answer to it. Some people are better in just conveying facts. Others are better in having the students working on it. So it'll come down to teaching style. And I don't know, in a decade or two, it'll be interesting to see what the teaching landscape looks like then. But I'm torn between that myself. Yes. Yes.
Yeah, great graphic visualization. Maybe a remark about the color. But I was struck by the similarity of a lot of things, the language, the critical threshold point, and so on. The similarities with exercise physiology.
Yes. Lots of the terminology is kind of stolen from the medical world, like the critical temperatures. If you look into any kind of animal physiology book, they'll describe critical temperatures for mammals as something very different when we start putting energy into actually thermoregulation. So it's a completely different concept, but someone liked that term and then applied it in the invertebrate world, which then confuses students and myself and others. So lots of their work then first starts in the mammalian world, and then invertebrate physiologists like me say, okay, what can we apply? That's why there's lots of overlap. That's why we always need to carefully define, okay, we are talking about this and not the other thing. Now, I do see it as an advantage of having worked in the mammalian and medical world for a while, because I read lots of the literature, which many people who go straight through the invertebrate world never see. And that's clearly allowed me to draw the different ideas from here and there. And so far it worked. Yes? I feared someone would ask that question.
Turn off the light.
There is this kind of the Bible on green crab biology is published in 1967, and that guy Crothers describes the green ones live more in the intertidal, where the algae are more green. The red ones live in the subtidal, where they're more red, they're better camouflaged. However, no, we find red and green ones in the intertidal. We find red and green ones in the subtidal, and they're red and green only at their belly, where the predators can see them. So it doesn't make any sense. And there are many other people describing what that could be, and to this day, no one has any good idea why they change color, whether there's any purpose. There are quite a few people who simply say it's some kind of metabolic end product which accumulates when they get older, which also can't be right, because if we cut off their eye stalks, and then we take away an important gland which regulates their molting, they turn towards molting way faster, and they still get red in a shorter period of time. So simple answer is, no one has any clue. Right. We probably need an organic chemist to figure out what that color actually might be. No, every single paper which I write about that stuff, I need to do this dance in describing this is happening and no one has any clue. And depending on what the reviewer believes, I get away with it or not. Yes?
Is there any evidence that the ones like Python and Notebook, sometimes they carrying a parasite load or
Yes.
carrying a hitchhiker?
Yes.
Any evidence whatsoever that some of that is coming or affects some to our ecosystem along with them and also affects their physiological tolerance?
That certainly is a possibility. And when I was setting up that work with those different populations, I needed main DMR here to inspect our system to make sure we don't release any of those hitchhiker species into our environment. They didn't worry about green crabs. They told me, they're here anyway, so why would we worry? They only cared about some cyanobacteria and other mean critters. That certainly could affect their physiology and their behavior. One of the parasites which I'm completely surprised that's not even here yet is actually a barnacle, a parasitic barnacle, which keeps all those green crabs in check in Europe. They settle on the green crabs, they grow into the green crabs and castrate them. And then they don't reproduce, and that keeps the population in check. That's why in Europe, green crabs are not a problem. And that parasite didn't make it over here yet, but it should be able to live in. Actually, people try to see whether we can use that here for getting rid of our green crabs, but they are not selective. They also affect rock crabs, they affect lobsters, and yeah, we don't want to let that one loose here. Yes, certainly a possibility. At this point, we don't know yet. Other questions?
Well, are you going to try to figure out that power question someday?
If Amy helps me. No, I don't know. I think this is really then coming down to a hardcore organic chemistry question, which is not my strength. I tend to do things which I can do. And if someone says, yes, here, we need to go for that, yes, sure. But that's not my first line of approach, because there's so many other questions. And with having this broader framework, now I can look at all those different aspects. I pick the ones which are cheap, or if I have the money, okay, whatever I can pay for, and what is kind of doable. There are many other things which would be fun to do, but it requires way more instrumentation or other expertise, and then I pick selectively. Yes, John. Right. Right. In a more controlled way, yes. That's kind of the holy grail of green crab research right now, because the Chinese love to eat green crabs when they're soft. They throw them in the oven, bake them, and then eat them. But they need to be soft, otherwise they're a little bit crunchy. And the problem is, there is no way of producing large numbers of soft-shell green crabs. And many people try to figure that out. And one of the approaches which we tried a few years ago was cutting off the eye stalks, which right behind the eye is an important gland which regulates molting. And every single textbook tells you that this gland releases a hormone which inhibits the molting. And if you take that hormone away, then there's that inhibition which is gone, and the animal molts, which just doesn't apply to green crabs. And we have no idea how they do it. They are just different. Once someone figures out how to make them molt in large numbers, then that person will be very rich. There were other attempts of getting rid of them by simply finding a way of marketing that food. We had with a collaborator who brought a guy from China over here who came up with an instrument to suck the meat out of crabs. It was literally a little suction tube, which you cut the crab open in the middle, and then you put the tube in there and suck that meat out, and it comes out at the other end as kind of individual muscle fibers, which you can then put in little crab cakes, and that works. And they used that in China on those Chinese mitten crabs, and we tested that thing here. Yes, it works on green crabs, but as long as people can make money on lobsters and rock crabs, why would they go through that tedious work of taking apart those green crabs? So in theory, yes, one could eat them, but here, why would you? Therefore, the idea is there, yeah, we can eat our way out of that problem. We just won't, because there are too many good alternatives. The most important question is, what is the food at the reception, right? It's crab cakes, of course. Here we go. Okay, thank you all for coming. I hope a few stay around for eating crab cakes. Thanks.